Good evening all, this is Sanjay doing second year MBBS at Madras Medical College. Today I am going to discuss about morphology and physiology of bacteria. First let us see morphology of bacteria. So bacteria, depending on their shape, are classified into cocci or spherical shaped, bacilli or rod shaped, and both cocci and bacilli are classified into gram-positive cocci, gram-negative cocci, gram-positive bacilli and gram-negative bacilli. Let us see some examples. This tabular column is important, it might be asked in MCQs. So if you take gram-positive cocci, this is actually an old edition. New edition, what we need to remember is, in clusters, Staphylococcus bacteria is arranged as a chain. It is arranged, Streptococcus is arranged like a chain, which is a gram-positive cocci. Then in pairs, it is arranged in pairs. Pneumococcus and Enterococcus are arranged in pairs. In tetra, it is Micrococcus. In octet, it is Sarcina. So, which is very important is this Staphylococcus, Streptococcus, and Pneumococcus and Enterococcus. These three, if you remember, it's enough for gram-positive cocci. So, gram-negative cocci, if we take, we will see the they are arranged like a pair. So lens shaped cells, bacteria such as meningococcus and kidney shaped bacteria such as gonococcus are arranged as pairs. If you take gram positive bacilli, in gram positive bacilli you should remember two important patterns like a chain. First one is, and then like uniform pattern. So example is Bacillus anthracis for chain arrangement. For cuneiform pattern or Chinese letter-like arrangement, best example is Corynebacterium diphtheriae. Next, if we take gram-negative bacilli, first we can take it comma-shaped, such as Vibrio cholerae, or you can take it as spirally coiled, such as spirochetes. We can take spirally coiled spirochetes, and then under gram-negative bacilli, you can take it as Vibrio cholerae, and you can take Haemophilus proteus. So these are all examples of gram-negative bacilli. Okay, so other spirally arranged like spirillum, spirochetes are also there. So first let us see about bacterial cell wall, properties of bacterial cell wall. See, bacterial cell wall is like a tough and rigid structure which protects the bacteria. It is around 10 to 25 nanometer in thickness and weighs about 20 to 25% of dry weight. Let us see the functions of the cell wall. So the cell wall provides protection, as I said earlier. Then it confers rigidity due to presence of peptidoglycan layer, and then it can protect a cell from the toxic substances, and also a bacterial cell contains endotoxin, which contain certain virulence factors that contribute to pathogenicity, and then immunity. So antibody raised against specific cell wall antigens or done on the cell wall, which will provide immunity. So very important is immunity, endotoxin, virulence factors like endotoxin, and then protection, rigidity, and protection from harmful or adverse chemical environment. Next, let us see about gram-positive cell wall. Okay, so cell wall of gram-positive bacteria is simpler in structure than that of gram-negative bacteria. This is very important point. Okay, so if you take peptidoglycan, each layer is a mucopeptide, that is murin chain. It is composed of alternating units of N-acetylmuric acid and N-acetylglucosamine molecules that cross-linked to each other, like we are tetrapeptide side chains and pentaglycine bridges, and then that is important point of peptidoglycan. So you can remember one point, or you can remember it is a tetrapeptide chain which is assembled from N-acetylmuramic acid molecule, which is composed of L-alanine, D-glutamine, L-lysine, D-alanine. So you can remember these 2 points are important, and then I'll show the diagram later. Then we will see the differences between gram-positive and gram-negative cell wall. So peptidoglycan layer is like a thicker. In gram-positive cell wall, you can remember positive, it is more higher, thicker. Negative, it is lesser, thinner. You can remember like that. And also you can remember directly the lipid content alone. Lipid content is present somewhat, very less in quantity. But in gram negative cell wall, it is opposite. It is present at a reasonable amount. If you take lipopolysaccharide, it is absent in gram. All lipid content lipopolysaccharide are very less or absent in gram positive cell wall, but in gram negative cell wall, it is present. Okay, so teichoic acid is present in gram positive, very important point. Teichoic acid is present in gram positive cell wall, and in gram negative cell wall it is absent. And also if you take aromatic amino acids, it is absent in gram positive cell wall and present in gram negative cell wall. Overall gram positive cell wall structure is simpler. Okay, it is simple than gram negative bacteria. Let us see in detail as we proceed. Next, let us see about peptidoglycan layer of gram positive cell wall. We know that there is a pentaglycine bridge. We have seen earlier that there are these molecules NAM and NAG are cross-linked to each other via tetrapeptide side chains and pentaglycine bridges. This is a tetrapeptide side chain. This is the pentaglycine bridge. So this diagram is very important to represent the peptidoglycan layer of gram positive cell wall. Okay, so that is what this is a brief summary about gram positive cell wall. If you take gram negative cell wall, the peptidoglycan layer is thin. Okay, that is you have to remember. And the pentaglycine bridge is absent. That is also an important point. And how it is linked is also important. The tetrapeptide side chains are directly linked, unlike gram positive cell wall in which pentaglycine bridge is present. In tetrapeptide side chains, they are directly linked to each other by the covalent linkage between D-alanine of one chain with mesodiaminopimelic acid of adjacent side chain. You can remember through this diagram. So between D-alanine and meso, so this bond D-alanine, this is meso diaminopimelic acid. So between these two chemical compounds, I will zoom it out. Between these two compounds, there is a covalent linkage, but here there is a pentaglycine bridge between D-alanine and L-lysine. That is important. The location is important. Next, I am going to cell membrane. So cell membrane of bacteria obeys fluid mosaic model, and then fluid obeys fluid mosaic model and contains phospholipid, lipid bilayer phospholipid, in which several proteins are embarrassed as integral proteins and peripheral proteins. Whereas in carbohydrates, some carbohydrates are often attached to the outer surface of plasma membrane. Carbohydrate cholesterol is also present. Next, function. So functions, it is a semi-permeable membrane acting as an osmotic barrier selectively, allowing particular ions and molecules to pass either into or out of the cell. So proteins and enzymes are present in cell. So in cytoplasmic matrix, cytoplasmic matrix is usually packed with ribosomes, storage granules called inclusions, and imagination of cell membrane called mesosome. And then, though they lack two cytoskeleton, but they still have cytoskeleton-like system of proteins, and they have protoplast. If we take ribosomes, they have 70S ribosomes. Each 70S unit consists of 50S and 30S subunits. So the polysomes are formed. Polysomes are nothing but ribosomes along with mRNA to form polysomes. So that is very important and help in protein synthesis. Okay, so if you take mesosomes, inclusion bodies is simple, you can learn. So mesosome helps in bacterial respiration instead of mitochondria. They are analogous to mitochondria of eukaryotes. They possess respiratory enzymes and are analogous to mitochondria of eukaryotes. They may be involved in cellular formation or chromosome replication during cell division. Next, let us see about nuclei. So they do not have two nuclei; they have a single chromosome. Okay, that is nucleoid. Okay, so bacteria will have as I said, single haploid chromosome comprising of supercoiled circular double-stranded DNA, and they have a linear chromosome. Some bacteria have linear chromosomes, some have two chromosomes. Like Vibrio cholerae has how many chromosomes? Two chromosomes. So bacterial DNA will divide by simple binary fission, and then bacteria contains extra chromosomal DNA that is outside nucleoid, that is called as plasmids. So it is important in gene transfer and in genetic engineering. Next, we have to see our capsule and slime layer. So outside the cell wall there is a amorphous material called glycocalyx. Okay, so the glycocalyx layer is very thick, very tight and tightly attached to the bacteria and is not easily washed off. It is called capsule. But if the glycocalyx layer is in the form of unorganized loose material which can be removed easily, it is called the slime layer. Okay, so examples are most of the bacteria capsules are polysaccharide in nature. Bacillus anthracis, the bacterial capsules are polypeptide in nature. So function of capsule is like protection from phagocytosis is very important, and then it will prevent the cell from drying or desiccation, and it protects the bacterium from the action of lysosome and bacteriophages and so on. Okay. So if you take flagella is an important component of bacteria which confers motility to bacteria. So let us see how flagella is arranged. So in arrangement of flagella, we have four categories: monotrichous, that is only single flagella at one end; lophotrichous, multiple flagella at one end; peritrichous, uh, flagella arranged surrounding the cell, like surrounding the cell at all direction. We have peritrichous, and then amphitrichous. And in amphitrichous type of arrangement, two flagella is present at the opposite poles. So there are four types of flagellar arrangement. So let us see the examples. For monotrichous, it is Vibrio cholerae and Pseudomonas. For lophotrichous, it is Spirillum, and for peritrichous, it is Escherichia coli. Okay, so in the latest edition is given like monotrichous type of arrangement of flagella is present in Vibrio cholerae, whereas peritrichous type of arrangement is present in Escherichia coli, and lophotrichous type of arrangement is present in Spirillum, and amphitrichous arrangement is present in Alcaligenes fecalis. Next, we will be seeing about ultrastructure of flagella. So flagella consists of filament, hook and basal body. So filament is the longest portion of flagellum, and it is a hollow rigid cylinder made up of a single protein that is called as flagellum. If you take basal body, it is the portion of flagellum which is embedded or inserted inside the cell. So it is made up of 2 to 4 rings connected to central rod. This is structure of basal body. Hook means it connects filaments, links the filament to the basal body. Next, we'll see about bacterial motility. Pili. Fimbria pili are short, fine hair-like appendages which helps bacteria to attach to other substances and hence called organ of adhesion. So pili are made up of protein called pilin. They are antigenic. However, antibodies against pilin antigens are not protective. Okay, so again they are not related to motility yet. That is very important in case of pili and fimbria. and can be found both in motile as well as non-motile organisms. So types of pylades: common pylades, simple pylades, they help in bacterial adhesion. Sex pylades, they help in bacterial conjugation. That's very important. Bacterial conjugation by forming conjugation tube through which the bacterial gene transfer takes place. Okay. So next one is uh, L form bacteria. These are atypical forms of bacteria. So if you take L form bacteria, uh, so the bacteria lose cell wall, they become spherical, whatever be the original shape. Irrespective of whatever be the original shape, if the bacteria lose cell wall, by default it becomes spherical shaped. So this will occur spontaneously or after exposure to penicillin or lysosome. So L form helps plays a vital role in persistence of polynephritis. If you take L forms of bacteria, like if we take mycoplasma, they too lack cell wall, and it is the stable form of stable L form bacteria. Okay, so that's it about L form. So it's easy to remember. So L forms are maintained only in presence. See, unstable L forms, you see, bacteria will lose the cell wall in presence of penicillin. Okay, so to resist itself against penicillin, the bacteria have to lose their cell wall. Okay, these cell forms are maintained only in presence of penicillin, and they can convert to original morphology. And see, if we remove penicillin, what happens is the resistance shown by the bacteria disappears, and hence again cell wall develops. So two types are there: protoplast and spheroplast. Protoplast are gram-positive bacteria whose cell wall is entirely removed. Gram-positive bacteria without cell wall. Spheroplast is gram-negative bacteria whose cell wall is partially removed. That is very important to be remembered. So L bacteria can come as short notes. Next, bacterial spores. So from innermost towards the outermost, the layers of spores are core, cortex, coat, exosporium. Core is the innermost part, contains the DNA material, and cortex and coat lie external to the core and separated from each other by a membrane, and the outermost layer is called as exosporium. So we have innermost layer core, then outer cortex, outer coat, and the outermost exosporium. So we have core, cortex, coat, exosporium. Sporulation. Sporulation means formation of spores in the early stage of bacteria. So during sporulation, the bacteria do not divide. Sporulation commences when growth ceases. So due to lack of nutrients, sporulation will occur. That is, the bacteria forms a protective cell wall which winds around itself, and numerous thousands of spores are formed. Though the bacteria do not divide, thousands of mature spores are formed, which is the deposition of calcium and dipicolinic acid into the spore cortex. So this calcium and dipicolinic acid will help the spores to be resistant against heat and disinfectant. So we learned 4 layers, very important, that is core, cortex, coat, and exosporium. Next, germination. So it is the conversion of dominant spores into active vegetative cells. Next, let us see about physiology of bacteria. So water is present in bacteria, about 80% of total bacterial cell. So the minimum nutritional requirements required for bacteria include carbon, nitrogen, hydrogen, oxygen, and some inorganic salts, or like sulfur, phosphorus, and other elements like sodium, potassium, magnesium. These are inorganic salts. Okay, now let us see about bacterial cell division. So bacteria divide by binary fission. So the cell division will start when a bacteria reaches a critical mass in its cellular contents. So nuclear division means the two strands of bacterial DNA are separated and then they themselves replicate to form new complementary strands. Thus, two identical double-stranded molecules are formed. In cytoplasmic division, we see the transverse septum grows across the cell from the cell membrane, following which the cell wall materials are deposited and the two daughter cells get separated. So daughter cells may remain partially detached even after cell division. Like uh, they may be arranged in pair or in chain like streptococci or in clusters staphylococci. Next, let us see the rate of multiplication in bacteria. So if you take Escherichia coli, it is 20 minutes. So generation time is required for bacterium to give rise to two daughter cells under optimum condition. We know that Escherichia coli generation time is 20 minutes. If you take Mycobacterium tuberculosis 10 to 15 hours, Mycobacterium leprae it is 12 to 13 days. So that is important. Then if you take bacterial count, we have two types of count, total count and viable count. So total count will indicates total number of bacteria alive or dead in the specimen. So counting the bacteria and viruses using counting chamber. Viable count means number of living cells. So let's see a bacterial growth curve. So bacterial growth curve is similar to the logistic growth curve which we studied in 11th and 12th. So when a bacterium is inserted in a suitable liquid culture medium incubator, its growth is observed as in graph. So the first phase is lag phase. C. This is a period between inoculation and beginning of multiplication of bacteria. After inoculation, bacteria do not start multiplying immediately, but they take some time to build up enzymes and metabolites. Okay, so bacteria increase in size due to accumulation of enzymes and metabolites, and they reach the maximum size at the end of lag phase. Lag phase is nothing but the preparatory phase. Log phase only bacteria starts dividing and divides exponentially, so that growth curve takes a shape of straight line. At this stage, the bacterium is smaller in size and biochemically active, and it is uniformly stained. So, since it is biochemical, it is the best stage to perform biochemical reactions, and it is uniformly stained. Okay, and this log phase is the best stage to perform the Gram staining. So it's very important, maybe asked in MCQs. So to perform the Gram stain, log phase of bacteria is the best stage. And in stationary phase, due to exhaustion of nutrients and accumulation of toxic products and autolytic enzymes, the bacterial growth stops completely after some time. So that is what it is needed. Then fourth phase is decline phase. So in this phase, there are four stages, four process. The bacterium becomes gram variable, and more storage granules are formed, and sporulation occurs in this phase. And we have seen one more process. Uh, in log phase, we have seen that gram staining is the best stage to perform gram staining is log phase. But here, sporulation occurs, and bacteria produces exotoxins, antibiotics, and these 4 points are important. And bacteriocins. Decline phase means the bacteria stops dividing completely. So cell death will occur due to accumulation of toxic products. So let us see some of the factors affecting growth of the bacteria. So you can remember oxygen. So there are obligate aerobes, facultative anaerobes, microaerophilic bacteria, obligate anaerobes, and aerotolerant anaerobes. So carbon dioxide. So organisms that require higher amount of carbon dioxide for growth are called capnophilic bacteria. So examples Streptococcus pneumoniae and Brucella abortus. Next, temperature. So most of the bacteria grow optimally at 37 degrees Celsius. However, so different species that optimum temperature varies. So for example, cyclophils, they grow at temperatures below 20 degrees Celsius. So example is mostly they are saprophytes. Example is Pseudomonas. And then next we will see about mesophils. Mesophils nothing but they grow within a temperature range of 25 to 40 degrees Celsius in between. That is a pathogenic bacteria, most of pathogenic bacteria. Thermophiles, they grow at higher temperature. range 55 to 80 degrees Celsius. Example, Geobacillus sterothermophilus. pH. So Vibrio cholerae is capable of growing at alkaline pH, and lactobacilli can grow at acidic pH. So it varies below pH 4 in acidic medium. So most of the pathogenic bacteria, however, grow between pH 7.2 to 7.6, which is same as that of blood pH. Next, light. So bacteria grow well in darkness, but they are sensitive to UV rays and other radiations in light. Then osmotic effect. So sudden exposure to hypertonic saline will cause plasmolysis, and exposure to distilled water may cause cell swelling and rupture plasmoptosis. These two terms are important. And then mechanical and sonic stresses. Due to vigorous shaking with glass beads, the bacterial cell walls may be ruptured, or by exposure to ultrasonic vibration also the bacterial cell wall gets ruptured. Moisture and desiccation. Moisture is needed for growth of bacteria because 80%, we have seen before, 80% of bacterial cell wall consists of water. However, the drying has various effects on different organisms. Okay, so that's it about this chapter. So the next video we will be dealing with laboratory diagnosis of bacterial infections. Thank you.